Antisense Technology. Antisense oligonucleotide and RNA interference. Antisense technology presents an opportunity to manipulate gene expression within cells to treat an endless number of diseases and is a powerful tool for studying gene function. The antisense approach utilizes antisense agents to fight various diseases by regulating the expression of a specific factor, the presence of which actually causes that particular gene. Some antisense approaches have evolved over the last few decades. Explicitly, the introduction of antisense oligonucleotides ASODN by Zemanick in the 70s, the description of ribozymes by Zek in 80s, and the first demonstration of short interfering RNA siRNA by Mello in the 1990s. Recently, microRNA miRNA replacement therapy has emerged as a new approach to treat human diseases like cancer and various neurodegenerative diseases. Replacement therapy involves the reintroduction of synthetic version of a natural miRNA, the microRNA, that gets depleted in the diseased tissue. Highly specific and effective gene silencing of any disease can be achieved by an accurate knowledge of target mRNA sequence and rational design of its complementary antisense agents for the down regulation of its protein message. Thus, these are being extensively explored for personalized therapy of cancer, HIV, and other mutating viral diseases like COVID-19. Gene silencing also has a great potential as a chemosensitizing agent to overcome the difficulties of drug resistance and dose limiting toxicities of chemotherapeutic agents. This technique differs from that used with conventional drugs in that it precisely checks the formation of disease causing proteins by down regulating its expression rather than relieving the symptoms of the disease after manifestation. Moreover, this technique can be differentiated from the genetic approach by its action on mRNA expressing the disease causing protein rather than acting on a particularly faulty gene. The success of this approach relies on understanding the correct sequence of RNA that carries the protein message responsible for the disease of interest. Fortunately, the completion of the Human Genome Project endows us with a rich source of information on target genes for the rational design of antisense drugs within hours for research and clinical trials. Antisense oligonucleotide formivircin is administered as a local injection to treat retinitis. Another similar approach to inhibit proteins is via specific three-dimensional complex structured molecules called aptamers. Pigatinib is an FDA-approved aptamer for the treatment of wet macular degeneration. This important class called aptamers. The evolution of antisense drug technology. Since its discovery, antisense technology has continued to progress rapidly. It was discovered in the 70s that the expression of a specific gene product could be inhibited using short complementary DNA sequence. This led to intensive research on the antisense approach. In 78, the concept of antisense technique came into view after the discovery of single-stranded DNA molecules known as antisense oligodeoxynucleotide AS oligonucleotide deoxynucleotide. Antisense oligodeoxynucleotide ASODN by Zemek and Stephenson. I repeat again, antisense oligodeoxynucleotide. Since then, new applications of antisense technology have continued to develop rapidly. In early developmental stages, blockage of target protein expression was achieved by administering the whole DNA or RNA locally at therapeutics. Then, single stranded DNA molecules, antisense oligonucleotides, were first locally administered for the treatment of tumors. Depending on the structure, ribozymes can degrade or modify the target mRNA to produce correct sequence. As oligonucleotide and ribozymes were already in clinical practice as genetic therapeutics long before small interfering RNAs were developed as potential medicinal agent. In 92, Pfizer were the first to describe microRNA as a mechanism of action of antisense oligonucleotide for the destruction of target mRNA. They are natural cellular defense mechanisms by virtue of which the presence of double stranded viral DNA triggers the mRNA degradation. Introduction of 20 to 23 nucleotide ring, short ring RNA, could exhibit antiviral activity by blocking the expression of viral proteins, which led to the progression of siRNA in therapeutics to block the production of disease related proteins. Since the discovery of antisense technology, there have been numerous advances both in the structure and properties of oligonucleotides used as therapeutic agents. Compared to that of whole DNA or RNA, RNAi is achieved using siRNA, double stranded RNA, and shRNA, short pin RNA. Several modifications such as novel bases, sugars, conjugates, and chimeric technology have been tried to improve the pharmacokinetic and pharmacodynamic properties of these oligonucleotides. Because the biological activity of antisense oligonucleotide at the site of action is dependent on factors such as its concentration, concentration of mRNA, rate of synthesis and degradation of mRNA, and the type of terminating mechanisms, various strategies have been employed to improve the properties of systemically administered oligonucleotides. Ribozymes have gained attention as antisense oligonucleotides. I repeat, ribozymes have gained more attention than antisense oligonucleotides. Ribozymes are enzymes that cleave single stranded regions of RNA by transesterification or hydrolysis of a phosphodiester bond. To achieve RNAi, either ribozyme encoding sequences are incorporated into plasmids or chemically modified minimum ribozyme structure is administered. Let us have a diagrammatic look of how it is administered, gains access within the cell, and then acts accordingly. A few of the limitations. Endothelial signs that line the vascular lumen present barrier to the antisense oligonucleotide based therapy, as oligonucleotides need to cross the endothelium before being delivered to the tissue parenchyma cells. The endothelial cells tightly adhere to the underlying extracellular matrix via integrins and to each other via several adhesion molecules, forming thus tight intracellular junctions with very small intercellular spaces. Since oligonucleotides travel across the endothelium via a paracellular route involving imperfections in these intracellular junctions. However, in certain tissues, such as those of liver and spleen, these endothelial intercellular spaces are relatively more extensive. Now let's have a look at the molecular mechanism of antisense oligonucleotides. Oligonucleotides bind to specific mRNA, thereby downregulating its expression and that of the encoded protein. However, the mechanism by which these oligonucleotides interact with the complementary mRNA and induce a biological effect are complex and difficult to elucidate completely. The ultimate goal of an antisense agent is to suppress or completely block the production of the related gene product. This means that in the process of transition from dRNA sequence to amino acid sequence, the normal transcription and translation apparatus must be affected. The formation of a protein product involves three distinct steps. In the first step, the sense strand of the DNA is transcribed into pre-mRNA. In the second step, the pre-mRNA is converted into a mature mRNA via the simultaneous action of three separate processes: capping, intron excision, and polyadenylation. Finally, the third step in the mRNA is transported to the ribosome for translation into appropriate polypeptide. Thus, antisense drugs can act by inhibiting any of these steps involved in the normal protein production. Although activation of RNA's H enzyme activity is thought to be the mechanism of action for the majority of antisense oligonucleotides, many still exert their biological effect via other reported mechanisms. What do you mean by aptamers? Aptamers can be considered chemical antibodies having the properties of nucleotide based therapies. These can be used to knock down the expression of target extracellular as well as cytoplasmic proteins. Aptamers are short stretches of RNA or DNA with a specific three-dimensional structure that can form complexes with the target protein to exhibit its expression by blocking its activity. It is not essential for the aptamer to be complementary to the target mRNA. Instead, its three-dimensional tertiary and quaternary structures determine the specificity and binding capabilities. Aptamers can be well utilized as non-immunogenic alternatives of antibodies and even at 1,000 times higher doses. Aptamers are acquiescent to the amendments that apply to the other nucleotide based antisense agents. Thus, aptamers can be tailored according to various required modifications while safeguarding their structure, specifically the binding region. Another closely related area is RNA interference, RNAi technology. RNAi was first discovered as a cellular protecting mechanism against invasion by foreign genes in Caenorhabditis elegans and subsequently observed in various eukaryotes such as insects, plants, fungi, and vertebrates. Interference RNAi is a powerful intracellular mechanism for sequence specific post transcriptional gene slicing initiated by double stranded RNA homologous to suppression of the gene. RNA interference. I repeat, RNA interference technology. Double stranded RNAs are processed by DICEA, a cellular ribonuclease 3, to generate duplexes of about 21 NAT with 3 overhangs called small interfering RNA, siRNA, that mediate sequence specific messenger RNA, mRNA, sequence specific messenger RNA degradation. The small interfering RNA, siRNA molecules in mammalian cells are capable of specifically silencing gene expression. Repeat, the small interfering RNA, siRNA, in mammalian cells are capable of specifically silencing gene expression. Thus, small interfering RNA, RNAi technology has been used as a new and powerful alternative to other gene silencing technologies such as antisense and ribosomes. Antisense technology is designed to deliver to the target cells antisense molecules that can hybridize and specifically inhibit the expression of pathogenic genes. In the past, the term antisense included several distinct but related approaches, including classical antisense or anticode, ribozyme, or catalytic ribonucleic acid triplex or antigene and aptamer technologies. A unified terminology has not yet been developed to describe technologies in this field. The first antisense drug, vitravine, has been approved for the treatment of patients with cytomegalovirus induced retinitis. Several antisense oligonucleotides have entered phase 1 to 3 clinical trials as anti cancer agents administered alone or in combination with conventional chemotherapy. Another closely related area is, I repeat again, RNA interference technology. First discovered as a cellular protective mechanism against invasion of foreign genes, we are repeating this, subsequently observed in various eukaryotes such as insects, plants, fungi, and vertebrates. Now let's have a look at the antisense technology. The normal way, DNA transcription, mRNA translation, and ultimately protein. Whereas in the antisense technology, this is sense. In the antisense, the microRNA, iRNA, interferes at this level and inhibits or blocks the amplification of certain deleterious products. So herein we have had a look. This is a presentation of gene expression process and mechanism of action of antisense oligonucleotides. Antisense oligonucleotide is complementary to the target RNA sequence. It specifically hybridizes with target RNA, resulting in translational arrest and increased RNA degradation by activating RNase H and or decreased RNA anabolism by inhibiting RNA splicing. So now we have the focus in this era of COVID-19 of using antisense technology in arresting viral replication. The antisense technology.